Please allow me to welcome you here for another MENDEL lecture. It's actually now more than 21 years we are running the MENDEL lectures. We have been able to acquire here really famous world science speakers, and today it's not going to be any different. So it's my great pleasure to actually introduce Johannes Walter, today's speaker. He was born in Germany, but then very early on he moved with his family to United States. He studied his bachelor degree at the University of California at Berkeley in biochemistry, followed with a Ph.D. at Yale University at the Department of Molecular Biophysics and Biochemistry, working on regulation of transcription in Drosophila, and then moved in 1995 to University of California in San Diego, joining John Newport, actually working on frog cell-free extract, where he actually showed for the first time a vertebrate DNA replication. And since 1999, he joined Howard University, where he is still now. In 2010, he was appointed a full professor, and in 2013 also investigator of Howard Hughes Medical Institute. Throughout his career, his lab is focusing, really utilizing the wonderful system of frog egg system, studying a molecular mechanism of not only DNA replication but also DNA repair, and also the diseases associated with these processes. Throughout his career, he has contributed really significantly in numerous areas, not only in replication, studying, let's say, a proteosomic pathway regulating or limiting the replication to occur only once per cell cycle, studying termination of replication, also characterizing the DNA cross-linking repair, also proteomic repair of protein DNA cross-links. Also a fundamental work in respect of characterization of engineering pathway and the combination of the cell-free system with a single molecule technique. And last but not least, it's also utilization of AI, AlphaFold, for screening a new protein-protein interaction and kind of discover a new interactions, but also ideas that could be testable. And that's what I hope we will hear to some extent today. But before I will ask him to present his lecture, I would also like to, and if you can join me, to congratulate him and award him the MENDEL medal for all of these achievements in science. So congratulations.
Thank you. Wow. Can you hear me? Okay, great. I have a really long list of things to thank Lumiere for. The first is the nice introduction. The second is the opportunity to give this named lecture. It's truly a deep, deep honor. Thank you for hosting a wonderful day of science at the university today. It was really interesting. I learned a lot of interesting things and what great science goes on here. And finally, thank you for introducing me to the jewel of your city, Brno. It's really a wonderful place. I think you're all very fortunate to live here. Okay, so Lumiere also asked me to say a few words about key people who dictated my path in science. And so really, I think the first and probably most important person to mention is my father, Johannes Walter. He was a biochemist. And he really showed me firsthand how wonderful it can be to pursue a life of science and follow your curiosity. My mother actually studied art, and she passed on a little bit of her artistic sensibilities to me, and I try to look at science through an artistic lens. I love a beautiful experiment. I love when it tells us something, but I also love when it looks really nice, and my students and postdocs know that. And I'd also like to thank, or certainly mention and thank, my postdoctoral advisor, John Newport, who I think had probably the biggest impact on the way I do science. He believed very deeply that most, if not all, biological mechanisms are elegant and logical and often simple, and he looked for those properties and qualities in any model that would describe natural phenomena. And that's a lens through which I try to also look at science. Okay, I'm trying to think if there's anything I forgot to say before I move into the science. I hope I got everything. So yeah, today I want to tell you how AI has really changed how my lab does science. And I also want to tell you how we are trying to push the limits of AI to be as informative and transformative as possible. So as Lumir mentioned, my lab has been very interested in mechanisms of DNA replication and DNA repair over the years, and we invariably find ourselves looking at different mechanisms, and when we do so, we almost always are essentially asking the question, what are the proteins involved and how do they interact? And I'm sure many of you here know how laborious and difficult answering that question can be using conventional approaches such as co-IPs and deletion analysis and mutagenesis and so forth. And so what I want to tell you about today is how we came to realize that AlphaFold is not only a very powerful way to determine the structure of proteins or complexes that we know exist, but also as a way to discover new protein-protein interactions. And so the first half of my talk will really be sort of a short story about a protein called Donsin and how our effort to understand what it does opened our eyes to the awesome power of AlphaFold and AlphaFold multimer. And then the second half of the talk will really deal with our efforts to push this approach to the limit. So to ask, can we take a new protein that we know almost nothing about, and can we actually use AlphaFold to screen it against the entire proteome to look for all of its functional and meaningful partners? Is it too loud for you? No? It's okay? All right. Okay, so this story that I want to tell you about has to do with DNA replication. So I want to give you a little bit of background about what we know about replication. So eukaryotic cells prepare for replication in the G1 phase of the cell cycle when a few proteins, ORC, CDC6, and CDT1, recruit pairs of MCM 2 through 7 ATPases to each origin of replication. These are the future helicases that will unwind DNA at the replication fork, but they are initially loaded in an inactive state as these head-to-head double hexamers that encircle both strands of the DNA duplex. And it's only when cells enter S phase and two helicase cofactors, CDC45 and GINs, are recruited to these MCM 2 through 7 double hexamers by two protein kinases, DDK, which stands for DBF4-dependent kinase, and CDK, which stands for cyclin-dependent kinase, that the full so-called CMG helicases are assembled. And here is a cryo-EM structure of the CMG helicase. Here you can see the ring of 6 related MCM ATPases. You can see the nice central channel that DNA goes through. And decorating the outside of the ring are those MCM ATPase co-activators, CDC45 and GINs. And as I said, initially when the CMGs are assembled, it's double-stranded DNA that goes through the central channel, and the final activation step for the helicase is that you have to expel or extrude one strand so that ultimately the CMG encircles just one strand of the DNA, and it then uses the power of ATP hydrolysis to translocate along that strand and pry apart the duplex. Once the helicases are active, they can unwind the origin, and then you can assemble two replisomes containing leading and lagging strand polymerases and various accessory factors. Okay, so as I said, today I want to tell you about the role of a protein called donsin in DNA replication. Now, donsin is mutated in a human condition called microcephalic dwarfism that is characterized by severely restricted pre- and postnatal growth, resulting in individuals of extremely short stature. And the reason why we became interested in donsin was because it was already known that mutations in various DNA replication factors cause this condition. These, of course, are hypomorphic mutations. Null mutations would be lethal. So not only factors involved in replication initiation, but also some polymerase subunits, as well as a few factors involved in the replication stress response. And given our interest in all of these features, we wondered whether donsin might also be involved in DNA replication, and we wanted to understand what role it might play. So, Yang Lim, a really outstanding graduate student, used our favorite experimental system, frog egg extracts, to try to understand what donsin does. So in this approach, we take female frogs, we inject them with hormone, it makes them ovulate, they lay thousands and thousands of eggs for us, and then we break open the eggs and essentially make a total egg lysate that contains the entire frog egg proteome, some 10,000 plus proteins. And what is really remarkable about these extracts is that you can add pretty much any DNA template into the extract, and the extract will then fully replicate the added DNA by mechanisms that, as far as we can tell, very, very accurately reflect what happens in a cell. And what's really important about these extracts is that they recapitulate processes like vertebrate DNA replication that have not been reconstituted with purified components. So it allows us to ask the question what a protein like donsin might do in DNA replication or the replication stress response. And I'm sure you all have already seen the result here, and that is that when we deplete donsin from the extract using antibodies, DNA replication was completely eliminated, and we could rescue that effect by adding back recombinant donsin. So that showed that, at least in this experimental system, donsin appears to be absolutely essential for DNA replication. Yang went on to ask what step in replication donsin might play a role in, so he repeated this experiment, but instead of measuring the incorporation of radioactive DATP to look at DNA synthesis, he recovered the plasmids out of the extract and used Western blotting to ask which factors are or are not attached. And you can see here that in the absence of donsin, the MCM 2 through 7 helicase is still recruited to the DNA, that licensing step that prepares origins for initiation is normal. However, the assembly of the CMG helicase, as measured by CDC45 and GIN's recruitment, was completely defective, okay? And so that result is depicted here, and I should point out that although in this experiment CDC45 loading was also defective based on some data from us and others that I don't have time to show you, we actually think that in the absence of Donson, CDC45 recruitment is basically normal, but if GIN's recruitment fails, then the association of CDC45 is so weak that it doesn't survive the chromatin recovery process, okay? So really, based on what I showed you and some things I didn't show you, we really think that Donson functions specifically at the GIN's recruitment step, that is, promoting its recruitment with the MCM CDC45 complex to form the final GIN's CDC45 MCM2 CMG helicase entity. And before I get into the mechanistic aspects here, I want to take a brief moment to point out that we think Donson has the same function in mammalian cells, and this is based on a very nice collaboration with Andrew Jackson's lab in Edinburgh, who received an auxin degron cell from Masato Kanamaki in Japan, and showed that if we arrest cells in the G1 phase of the cell cycle and then add auxin to degrade Donson, then if we release those cells into S phase, then when Donson is absent, there is absolutely no evidence of any DNA synthesis occurring. And if you isolate the chromatin, you can see that again, the CMG assembly is defective. So that really argues that this is a conserved role of Donson in vertebrate cells. And I would also like to point out that they engineered a patient-derived point mutation, that is a mutation that causes dwarfism in humans, and found that in such cells that express biallelic dwarfism mutations, CMG assembly is also reduced. It's not fully absent, of course, that would be incompatible with cell proliferation, but it seems to be significantly reduced, and that suggests that the dwarfism phenotype itself might be due to defective or partially defective CMG assembly. Okay, so now onward to the question of what Donson actually does. So how does it promote the association of GINs with this MCM CDC45 complex? And from work by a lot of other groups, it was already known that this recruitment step depends on an accessory factor called TOP-EP1 and, at least in yeast, a non-catalytic function of the leading strand polymerase pol epsilon. So Yang was working very hard on trying to understand what Donson does using all of the conventional approaches, and he wasn't getting very far. And this was really around the time when the whole AlphaFold revolution exploded around us. And hopefully everybody is aware of AlphaFold, but just in case, I will point out that it is a deep learning AI system that can take an amino acid sequence, consider genetic or evolutionary relationships with related proteins and any structural information that is already in the PDB, and then make, with very high accuracy, very compelling predictions about the protein's structure. So the first thing that Yang did was, and I should point out here, and it'll become relevant in a moment, that AlphaFold was trained using 5 distinct regimens, and so when you give AlphaFold a protein, it will make up to 5 predictions, okay, and that'll become relevant in a moment. But when Yang looked at the AlphaFold prediction for Donson, it wasn't very informative. It just suggested that it has a long, unstructured N-terminal tail, a globular domain, and then a loop that protrudes off of the globular domain. So that didn't really tell us much of anything about what Donson is doing. But based on our experience in studying molecular mechanisms, the key to how a protein like this would work would really be connected to its partners. So what are the other proteins that it binds to? And so in the spring of 2022, a version of AlphaFold called AlphaFold Multimer was released by the DeepMind group and published here in bioRxiv as a preprint. And it uses very similar principles as AlphaFold to make structure predictions, but it was trained on multimeric protein complexes. And indeed, it's very successful in making predictions about binary or even ternary or quaternary structures, as many amino acids as you can fit into 3,000 or 4,000 total amino acids. And here's just an example of two subunits of the GINS complex, and you can see that the AlphaFold Multimer prediction matches very, very well the experimentally determined structure. Now, one very, very nice and absolutely critical feature of AlphaFold is that it not only makes a prediction, it also tells you how confident it is about that prediction. And so it puts out various so-called confidence metrics. One of them that is very popular is the Interface Predicted Template Modeling Score, or IPTM, which is on a scale of 0 to 1, with anything over 0.5 being considered a high-quality prediction. And you can think of this as sort of an overall or a measure of AlphaFold's confidence in the overall structure, okay, that it is predicting. Now, remember I mentioned that AlphaFold was trained with 5 distinct regimens, and so when you give it, in this case, a protein pair, it will make up to 5 independent predictions, and those are shown here. And if you overlay them, you can see that except for some of the floppy bits, it agrees, they all agree very, very well. And we have found that the agreement among those 5 different models is one of the best measures of confidence. And so what we did was to come up with a new and admittedly very simple confidence metric, which just quantifies the agreement among the 5 models, and we call that the average model score, okay? All right, so the reason I'm telling you all of this is because Ernst Schmidt, another terrific graduate student in the lab, who has a knack for computational biology, said, let's take Donson and let's use AlphaFold to predict, AlphaFold Multimer, to predict its structure with all 70 of the core DNA replication proteins that are known to be necessary and sufficient to support replication in a yeast cell-free system developed by the Diffley lab. And so he developed a folding pipeline where we rent GPUs in the cloud. We use a streamlined version of AlphaFold Multimer called CoLabFold to fold Donson with all 70 of these pairs to get all of these predictions. And then he has an analysis pipeline to calculate the various confidence metrics that I just told you about. And when we got the results from this, it was really very, very striking, because when we ranked the hits by that average models metric, the top 5 hits were all proteins that had already been implicated in the recruitment of GINS to the MCM 2 through 7 CDC45 complex. So the very step that we had shown is regulated by Donson, okay? So that includes Donson itself, suggesting that it homodimerizes, a subunit of DNA polymerase epsilon, one of the subunits of the MCM 2 through 7 complex, one of the subunits of GINS, and that top EP1 accessory factor, okay? But now the real beauty of AlphaFold is, of course, that it gives you an atomic level resolution prediction. And the striking thing was that when we looked at all of these predictions, they were predicted, all of those 5 partners were predicted to bind to 5 distinct regions in the Donson protein. So the extreme N-terminus was predicted to interact with one of the subunits of the tetrameric GINS complex. A more internal, very short peptide of 5 or 6 amino acids was predicted to dock on to Pol epsilon. This loop here that protrudes off of the globular domain was predicted to alpha helices on that loop are predicted to nestle into this little cleft here in MCM3. This part of the globular domain was predicted to interact with the BRCT3 domain of top EP1, and then a different part of the globular domain was predicted to mediate the homodimerization, okay? So that was kind of remarkable, but when we combined all of the predictions into a single picture, a potential mechanism of how Donson might recruit GINs literally leapt off the page, and that was that Donson would use its 2 short peptides here and here, and this part here, to organize GINs, Pol epsilon, and top EP1 into what we now call a preloading complex that is somewhat analogous to a similar complex that has been described in yeast, and that this loop here that protrudes off of the globular domain would then chaperone this preloading complex onto the MCM 2 through 7 CDC45 complex to deliver GINs to its proper location. And the really striking thing was that when we put all of these together by folding multiple subunits together and then used the binary prediction between Donson and MCM3 to dock this whole complex onto here, it placed GINs precisely in a location that was exactly where GINs is known to reside in the experimental CMG structure. So all of that together really suggested that Donson is a chaperone that delivers GINs to its proper location. Okay, that's all great, but of course we have to test these models, and because this is all published, I'm going to show you one example of how we did that. And of course, the approach was to perform site-directed mutagenesis at the predicted interfaces and then ask what are the consequences of that. So I'll just show you one example where we mutated an asparagine in Donson here at this interface between Donson and top EP1, and we took that mutant and we dropped it into egg extracts and then we pulled it out again and did Western blotting. And you can see that unlike the wild-type Donson, which co-IPs GINs, top EP1, and Pol epsilon, this point mutant still interacts with GINs and Pol epsilon, but it selectively loses the interaction with top EP1. And that mutant is almost completely devoid of replication activity, and it is also severely compromised for recruitment of the GINs complex to chromatin. Yang went through and he mutated all of the other interfaces, and they had the expected effects on DNA replication, providing very compelling support in the absence of any experimental structure determination for this model, that Donson is a chaperone that delivers GINs to its proper location. Now, I want to actually just take a brief moment and point out that 2 of these interaction points between Donson and its partners involve very, very short peptide motifs that are embedded within an otherwise unstructured region. So here it's about a 15 amino acid peptide. Here it's maybe a 6 or 7, or actually I think 5 or 6 amino acid peptide. And it's really remarkable that alpha-fold multimer was able to pick up even such limited interactions, and I think it bodes well for the prospect of using alpha-fold multimer to characterize what is probably a huge universe of sort of motif-based interactions. Now, I also want to point out that working contemporaneously Two other groups, Joe Yiel's working with Karim Labib, and then Alessandro Costa working with Aga Gambus, solved some structures of Donson with CMG and GINs in worm and in Xenopus, and although they don't cover the entire space of the structure prediction, they do agree very, very well with the prediction, or I should say the prediction agrees very, very well with the structure. I should also point out that a couple of other groups came to similar conclusions. So there's I think a very strong consensus now in the field that Donson is an essential new metazoan replication factor that is absolutely crucial to assemble the replicative DNA helicase. And we actually know, based on the work from Karim Labib's lab, that this concept extends at least to worms. And then I think this is a reminder to myself to point out for the aficionados and experts in the audience who actually know something about DNA replication that what I've depicted here is something of an oversimplification, and I'm happy to go into a little bit more detail in the Q&A on some of the subtleties, or I shouldn't say subtleties, additional aspects such as the actual importance and role for Donson dimerization, which you will recall was one of the top predictions. Okay, so that was really kind of the almost religious experience that we had that converted us to the just extraordinary power of AlphaFold multimer and its ability to just accelerate mechanistic discovery beyond our wildest dreams. And then we started kind of dreaming a little further and wondering whether this approach could be used sort of ab initio to discover the function of a protein that we know, let's say in the extreme, nothing about, right? The Donson case was kind of special in that we knew a lot about what Donson does. We knew it's required for replication. We even knew the step that it controls, and so therefore the scope of the in silico screen that we did to discover its function was really quite limited. But imagine that you're working on a protein, let's say you pull it out of a genetic screen or something, and you know nothing about what it does. Could you use this in silico screening approach without ever picking up a pipette to figure out what a protein does, okay? And so, since we already knew what some of the functional partners of Donson are, we thought we would kind of perform this experiment in the context of Donson. So in other words, would AlphaFold multimer, this in silico screening approach, would it have found Donson's partners if we folded Donson with all 20,000 human proteins, okay? And now to just kind of set your expectation level here a little bit, I'll say a few words about the human interactome. So there are about 20,000 proteins, which means that there are 200 million theoretical pairings between those 20,000 proteins. And current estimates, which are undoubtedly very, very inaccurate or approximate, suggest that maybe 1 in 200 of these is actually functionally relevant. And what that implies is that every protein has maybe on the order of 50 relevant partners, and that means in turn that if you do a proteome-wide screen, then only 1 in 400 of the proteins that you interrogate is actually going to be a correct hit. And the implication of that is, of course, that whatever means you use to score and evaluate your hits has to have an extraordinary level of specificity, okay? Even a very, very low false positive rate would confound the analysis and basically mean that most of what you're looking at is junk. So we did the experiment. We folded Donson with all 20,000 human proteins, and then we ranked the hits based on these existing confidence metrics that I told you about, right? The interphase predicted template modeling score, and then this metric that quantifies the agreement among the 5 models. And so what we're doing is we're just ranking the 20,000 hits according to the score, and you can see that when we do this with IPTM, the 5 functionally relevant Donson partners, Pol Epsilon Donson, top EB1 MCM3, and SLD5 are spread over the top 1,000 hits. If we do the same for average models, it's a little better. They're spread over the top 400 or 500 hits, okay? So clearly, these metrics are able to enrich for the proper targets, but I think you would agree that if we knew nothing about what Donson does, this wouldn't be extremely useful, right? Because we would literally have to go through hundreds of candidates to find the right guys. So what we need then is a better metric or algorithm, or I'm going to call it a classifier, that can tell the difference between a true prediction and sort of a spurious prediction made by AlphaFold. And this is, of course, a classic machine learning problem, right? So what we want to do is we want to train an algorithm, or what we call a classifier, to tell the difference between good and bad predictions. And to do that, to train a classifier, you need a very high quality sort of ground truth data set. In this case, we need a large number of AlphaFold predictions between 2 pairs, or between a pair of proteins that we know are true, or where the vast majority are true. And then we need the complementary data, which is a large group of predictions that are true, okay? So for the true negatives, we followed the idea that I just told you about, which is that if you randomly pair proteins from the proteome, the vast majority, greater than 99%, are almost certainly going to be false, okay? So that was pretty easy. For the true positives, we went back and forth between different ideas. Of course, we considered taking protein complexes from the PDB, but we worried that if we did that, then we would be biasing towards really well-behaved protein complexes that are of current interest to the community. So instead, we decided to dive into the growing number of cross-linking mass spectrometry data sets that are out there, where people take cells or cell lysates, treat them with cross-linking agents, and then use mass spectrometry to find peptides from protein pairs that actually interact with each other under those conditions, okay? But we went one step further, and that was that we took the pairs from those cross-linking mass spec data sets, we folded them with AlphaFold, and then we looked at the structure prediction and chose only those predictions where the amino acids that participate in the crosslink are actually close to each other in the prediction. And we reasoned that in that way, we could really enrich for a set of very high quality interactions that are likely to be true. Okay, so now we have our set of true positives and true negatives. We mix them together, keeping track, of course, of what's what. And now we want to train an algorithm that can separate them out again, right? And learn to tell the difference. And so in order to do that, we have to identify as many features of each pair as we possibly can, right, to instruct the algorithm. So we began with what we call structural features. So these include not only the classic confidence outputs from AlphaFold, but also measurable properties of the predicted protein complex, right? So we looked at the interface in an automated way, of course, and looked for the number of contacts between the proteins, the number of salt bridges, hydrogen bonds, buried surface area, anything that we could measure, right? And we fed it to this algorithm. And I won't show you the data, but I can tell you that when we did that, the resulting classifier that we trained was quite good. It was better than everything we had used before, but it wasn't quite where we wanted it to be. So we then, in addition to those structural features, we also used what we call biological features, okay? So we looked at the RNA-seq database. So we asked for each protein pair that we're looking at, are the genes that encode those proteins co-expressed in time and space? We asked whether the proteins are co-localized in the cell. We asked whether the genes encoding those proteins exhibit any codependencies in the cancer depth map or in the 1,000 or so CRISPR screens that are available online, and so on and so forth. And altogether, we identified 65 of these structural and biological features, and we used them to train a classifier using a machine learning approach called random forest, which I won't trouble you with further. But the idea is that you take 75% of your curated data set, of your positives and negatives, and you then ask this machine learning tool to use these features to separate these two groups as best you possibly can. And that involves giving each pair a score on a scale from 0 to 1, okay? So you can do that, and then you have your algorithm, and then the question is, of course, how good is it, right? So to test or measure performance, you take the 25% of the curated set where you know what the answer is, and then you use your classifier to look at each pair and give it a score, and then see how well did you do, okay? And of course, the dream scenario, a perfect classifier, would be one where above some score, all of the pairs that you call true are in fact true, and there are no false positives, and everything below that threshold would be a negative, okay? In the real world, with very complex data, such as these structure predictions, that is not how things go. And so really to measure performance, we have to look at these so-called receiver operator characteristic curves. And I will just spend a brief moment on this, mostly for any sort of computational specialists here, and I will then very rapidly move on to a more intuitive measure of performance. But just suffice it to say that what we're doing here is plotting the true positive rate versus the false positive rate as a function of the classifier score. So we start at the top right at the very lowest threshold, so a classifier score of 0, and then we ask what happens to the true positive and false positive rate as we increase the stringency, as we keep increasing the threshold of that score. And for a very good classifier, what you see is that the curve sort of tends towards the top left of the graph, and that's because what you can see is that at this classifier score here, we're still identifying we are still identifying the vast majority of the true positive pairs while including very low fraction of the false positives, okay? And I hope you can see from the shape of the curve that the classifier that we just trained is doing much better than these prior confidence metrics, okay? And another sort of single number that describes or quantifies the performance of a classifier like this is the so-called area under a curve. 1 being perfect, right? That would be a classifier that just stays all the way up the top and then crashes. Ours does very, very well. It's a 0.96. And the next best is this average models metric of 0.86. Okay, so before I give you a more intuitive demonstration of performance, I want to pause here and just say that on the basis of this and other things, we decided to call this classifier SPOC, that stands for Structure, Prediction, and Omics Classifier. And we quite like this because it finally explains hand gesture, right? Which clearly says these two proteins interact. Okay. But the reason, why we set out on this whole sort of escapade was to see if we can successfully rank true pairs in a proteome-wide screen. And this is the data that I showed you before, right, where we used either IPTM or average models to rank those 20,000 Donson pairs, and where we found that the 5 relevant ones were spread over the top hundreds of hits. So now if we rank those 20,000 predictions using the classifier, we get a very dramatic and very satisfying result, which is that the 5 pairs are in the top 7 out of 20,000. And I will immediately point out to you that Donson was not in the training set, okay? So there was no way for our classifier to be able to cheat. And I will also point out that we didn't keep doing this and tweaking it until we got this result, okay? And some of you will have noticed that there is this little dot here, and that is a new hit that is at position number 2, and it's a little too early to talk about it. What I will tell you is that when we finally did the IP mass spectrometry with Donson, the 5 known partners were highly abundantly enriched, and this new hit was also one of the very top hits. So that was a nice confirmation that this classification tool is working. But you would be right to be very skeptical, to be convinced here for an n of 1. And so what we did is we repeated this kind of ranking experiment about 20 times. Turns out that it's kind of expensive to fold 20,000 pairs, so we scaled it back a little bit. And what we did is we took pairs of proteins like this SLF1, SMC6 complex. The details don't matter, but it is a protein pair that we knew from the literature is a functional complex based on biochemical and genetic and or cell biological data, but for which there's no PDB structure, so that AlphaFold cannot cheat in any way. And then we took one of the 2 proteins in the pair, in this case SLF1, and folded it with 1,000 random proteins, mixed them all together, and then ranked them based on the different metrics. And here you can see that when we do this, IPTM puts the SMC6 correct partner at rank 87, average models puts it at 26. SPOC, the classifier, puts it at number 2, and initially we were disappointed, but then we realized that actually by random chance, a known partner of SLF1 had ended up in the 1,000 random guys, and that protein ended up at position number 1, which was kind of cute and satisfying. So we repeated this 19 times and quantified the results, and over these 19 experiments, the SPOC classifier places the correct pair at median rank number 1 out of the 1,000 or 1,001 pairs. The mean rank is about 2, and then the other metrics do progressively worse. You might say, oh, gee, average models is, actually doing really quite well, but remember that we did this on a scale of 1,000 proteins, so if one wants to think about proteome-wide screens, one has to multiply these results by 20. And then you can see that on average then, the average models would place the true partner somewhere between the 100th and 200th position. And I would like to point out also that in this group of 19 so-called mixing experiments, most of them were protein pairs that come from the genome maintenance field, but we did identify a few that were in a completely different pathway, and in some cases even totally different compartments, and the Spock classifier still worked really well. And that's not surprising because actually our training sets were completely pathway and compartment agnostic. Okay? All right. So I think that gives us some confidence that we now have a classifier that, in principle, can make sense of a proteome-wide in silico screen. And the dream scenario now would be to fold all 20,000 human proteins with each other and generate a comprehensive structural interactome. Now, the reason why we would really like to do that is because of the 1 million or so estimated actual protein-protein interactions, current estimates are that we know only about 50,000, okay? Only about 5% of those. And of those, only maybe 10,000 are structurally resolved. So that would be 1% of the total number of protein-protein interactions. So that means that the vast majority of PPIs are unknown, and a staggering 99% or so are structurally unresolved, right? So that is actually pretty shocking, and it suggests that there is just a gigantic space of structural and mechanistic biology that is currently inaccessible to us. All right, now, having said all of that, I have to give you the bad news, and that is that doing this would cost $80 million in cloud computing time, all right, which goes a little bit beyond my lab budget. So we decided to pursue a little bit more modest of a goal, and that was to fold 300 or so proteins that were near and dear to our hearts, that is, proteins involved in genome maintenance. So all the proteins known to be involved in replication, cohesion, checkpoint signaling, telomere structure and function, and all of the canonical DNA repair pathways. So that works out to about 40,000 pairs and comes with a much more affordable price tag, and we call this pathway analysis. So we did this, and this is what we call the matrix. So here are the 285 proteins arrayed across the top and across the side, and the intersection of every row and column is a protein pair. The colors designate the confidence that AlphaFold has in the prediction, and you can already see that there's a vast number of protein interactions predicted, and I think I took a screenshot here of the matrix without any sort of classification or any threshold applied. And what we've done is we've actually allowed people to sort the data based on their favorite classifier. As of yesterday, actually the Spock score is on there, so you can filter this data by Spock score, and you can adjust the threshold level that you want, that you feel comfortable, and you can sort the data that way. Of course, you can click on any one of these protein pairs and then retrieve confidence metric information, basic information from UniProt, but most importantly, an interactive structure reviewer that shows you what the structure actually looks like. You can download it here. We automatically actually search the PDB for each pair to see whether that protein complex has already been solved by experimental means, and if it has, you can retrieve it from the PDB and then superimpose it onto the prediction to see whether essentially what you're looking at is something that is already known, and you can move on to the next pair. We also load up these infamous predicted alignment error plots and PLDDT plots and so forth. I won't trouble you with the details of that. But really the intention here is to allow people to very quickly assess and triage thousands and thousands of protein interactions and look at them in this case in the genome maintenance pathway, but the hope is that others will apply this to other biological pathways and that this will become sort of a standard way of mining for new protein-protein interactions. Now you can also type in your favorite protein and get a rank list of the predicted partners, and so here you can see the usual suspects at the top of the list. Here actually we're sorting by Spock score. And actually as of yesterday, what we've done to give the community wide access to this powerful new classification tool is we've actually put this on the website. So if you have generated your own AlphaFold predictions, you can load them, just drop them here into this field, and then a couple of minutes later you will get an email with the Spock score. Okay, and the details of how to do that are on the website. Okay, now a very relevant question is, of course, what is a meaningful Spock score? And that's a kind of a complicated thing that we go into a lot of detail on in the bioRxiv paper that should have been out today, but isn't yet. I don't know what they're doing over at bioRxiv, but it used to take like half a day to get it approved, and it's been 3 days. I don't know what's going on, but it will be up there soon. But in order to give you some idea of what is a reasonable Spock score, what I've done here is taken two curated sets of structure predictions, ones that we know are true and ones that we know are false, and sorted them by Spock score. So the ones that are true, this is now actually a set of predictions that correspond to proteins in the PDB, okay, so where we know that they interact and we know there's a structure, but we chose ones that were deposited in the PDB after the AlphaFold training date cutoff. And then for the negatives, we again just used the randomly paired guys. And you can see that as expected, the random ones that should be largely negative are highly enriched for very low Spock scores. The ones from the PDB that are positive are highly enriched for higher Spock scores. But I want to point out, and this is a note of caution, is that there are actually a non-negligible number of pairs that we know are true because they're in the PDB that get a really miserable Spock score. So if you get a bad Spock score for your prediction, don't despair. It may be a false negative, okay? But we think that 0.3 is a pretty reasonable sort of cutoff because above this value, 88% of the positives are retrieved and only 2% of the negatives are included. Okay. Now, if you actually apply this criterion to all 40,000 of these predictions in the matrix, then what you see is that about 2,000 pairs have a Spock score greater than 0.3. So that would work out to about 7 partners on average for each of the 300 proteins in the genome maintenance space that we included in this matrix. And I will also point out one very important thing, and that is that 250 of these 2,000 pairs that have a very good Spock score are completely absent from the string database. Many of you know that string is a database that mines all possible data to look for functional and physical associations of proteins. And so at least 10% of the things that we are finding have a very good Spock score are completely off the grid, okay, as far as the scientific literature is concerned. So I think that suggests that if we really applied this comprehensively, we would be learning about a lot of new protein-protein interactions. Okay, now and I'm almost finished, don't worry. So the point of all of this, right, is to generate new hypotheses, right, that you're going to test. And I have spent hours and hours and hours gazing at the matrix. My wife complains that I have a new mistress called AlphaFold sometimes. And so I'll just give you one example. So if, for example, you type in DNA polymerase alpha, this is the DNA polymerase that primes Okazaki fragments during lagging strand synthesis. Then you get a list here, they're sorted by Spock score, and a whole lot of these are expected partners, but there are some very interesting surprises. For example, the large subunit of Pol alpha is predicted with very high confidence, super Spock score, to interact with a subunit of another DNA polymerase called Pol delta, which actually synthesizes the Okazaki fragments. And in addition, there's another very strong prediction that Pol alpha interacts with a protein called RFC1, which is a molecular machine that loads a ring-shaped molecule called pCNA that confers processivity onto this Pol delta polymerase. And if you look at the actual predictions, Pol D3 is predicted to use this beta hairpin to complete this beta sheet, and that's conserved from humans down to budding yeast. Interestingly, this subunit of the RFC clamp loader complex is predicted to interact in a very similar region as Pol D3, again, totally conserved among eukaryotes. And seeing this, I said, oh, well, let me fold all 3 of these together. And when you do that, something very interesting happens, and that is that Pol D3 interacts pretty much still in exactly the same way, but RFC now drapes over this beta, this composite beta sheet between Pol alpha and Pol delta, okay, suggesting that maybe the recruitment of RFC could be dependent on Pol delta. And if you think about this a little bit and put it all together, it actually suggests a very interesting model for how Okazaki fragment processing might take. So the idea is that even as Pol alpha is making a primer based on these AlphaFold predictions, that would suggest that already these 2 other machines are already tethered as the primer is being made. And then when Pol alpha lets go, when it's finished making the primer, RFC is already present and should be able to immediately engage with the primer to load pCNA, and then when RFC leaves, Pol delta is already present and can engage and extend the primer. And I should point out that actually single molecule imaging from Antoine Van Ooyen's lab from some number of years ago actually suggests that Pol delta, a single molecule of Pol delta remains associated with the replosome through multiple cycles of Okazaki fragment synthesis, in good agreement with this whole concept. Okay, so in summary, I hope I've convinced you that in silico screening using AlphaFold multimer is a promising new approach to discover functionally relevant new protein-protein interactions, and I want to point out right here that we're not doing this in a vacuum. There are several other groups that have realized the power of this and are pursuing similar approaches. What we have done, which I think is a little bit unique, is to train this powerful new classifier that can tell the difference between positive and negative interactions or predictions, and really makes it feasible to take this approach to a proteome-wide scale. We folded our favorite group of proteins and put the data freely available at a website called Predictomes, simply because there are way too many predictions, interesting predictions for any one lab to pursue, and we thought it was the right thing to put it out there to catalyze as much science as possible. And I told you about Donson, this very interesting and fundamentally important new vertebrate replication factor. Now, I do want to end on a note of caution and point out that this is not the solution to everything, and that there are some limitations of this approach. And one is the fact that there are clearly false negatives. So for reasons that we and others are thinking about, and I'm happy to discuss, there are definitely cases where AlphaFold simply fails to make a prediction, even for a complex that we know is true, and the fraction of those is actually pretty high. There's something called paralog confusion, where AlphaFold has a hard time telling the difference between different paralogs. So for example, if you fold all the MCM 2 through 7 related ATPases with each other, they normally assemble in a perfectly unique sequence. AlphaFold gets confused about who binds to whom, presumably because they're structurally and evolutionarily quite related. AlphaFold makes mistakes in where it places individual amino acids, secondary structure motifs, and even domains, and those errors are going to be compounded if you try to build a large protein complex from binary or ternary predictions. It is limited in its ability to accurately predict conformational dynamics, although people are working on addressing that, and at least based on publicly available versions of AlphaFold, it currently cannot model non-protein ligands, which is obviously a huge gap, but anyone who saw the press release from DeepMind in December knows that they claim to have solved that, but they have not made it available yet. We're hoping that that will become available soon. So all of this together means that experimental structure determination remains absolutely essential, and I think will for the foreseeable future. But all of those caveats notwithstanding, I think we are at a very exciting place where we have the, I think, potential, at least, to really start thinking about a comprehensive structural interactome. Once that gets done by someone with very deep pockets, and if one combines it with the kind of classification tool that I talked about, I think we could end up with a reasonable first draft of an interactome that will probably reveal just countless new functionally meaningful protein-protein interactions that could really unleash, I think, a new sort of revolution in mechanistic biology. Obviously, it could explain disease mutations and of course also accelerate drug discovery. So I want to stop here and thank the really terrific people who did the work. So Yang did all the biochemistry with some help from Olga and Rian. Ernst really drove the computational biology. I want to thank our terrific collaborators, my funding sources, and I also want to thank all of the Walter Lab members, past and present, who are just a really extraordinary group of people, and it's been just an amazing privilege to do science with them. So I will stop there and be very happy to take your questions.
Okay, thank you, Johannes, for fantastic lecture. Not only sharing the data with us, but here at the presentation or lecture, but also making it available for researchers. I think we are experiencing kind of revolutionary era at this point, and I think this will really spark a lot of interesting research using this tool or the tools which will become. So it's not me, now it's time for you and your questions for those who are interested.
Yeah, thank you, Johannes. Really great talk, as always. I have two questions. So one is regarding the Donson, because it creates such a nice compact structure with the top BP1, pol epsilon, and GINs. So how then this structure recognize the MCM with CDC45? Is it through some phosphorylation, and is it Donson recognizing this phosphorylation, or is it the top BP1?
Right. So I anticipated that question from you. So a couple of things to point out. One is that, and I glossed over this, in co-IPs, we have never been able to co-IP Donson with an MCM 2 through 7 complex, even though I told you there's the loop that protrudes off of Donson that is predicted to interact with MCM3. And what we think is that the way this works is that top BP1, which as you know, involves multiple BRCT repeats, including 2 at the N-terminus that interact with phosphorylated treslin. So we actually believe that the preloading complex is initially brought to the MCM double hexamer via this interaction, and that then brings Donson close enough and raises the local concentration of Donson enough so that it now can productively interact with MCM3. So that's how we're thinking about it. I'm not sure if I answered all of your questions.
Yes. And the second question was, so what do you think about later function of the Donson? Is it traveling with the fork? Can it have some role in the fork stability or maybe dormant origin firing later on?
Right. That's a great question. I don't think we know the answer, but a really beautiful study was just published from my ex-postdoc, George Chistol, who's at Stanford, and he did single molecule imaging in frog egg extracts, and he fluorescently labeled a whole bunch of factors, including Donson, and he showed that Donson loads, and he beautifully showed that actually it delivers 2 copies of GINs, and then it leaves. So before the origin actually fires, Donson is gone, and that appears to be possibly coupled to the action of RecQL4, which is another factor, as you know, that probably functions late in origin firing.
Okay, thank you.
Hi, it was a great talk. I was wondering what is the function of dimerization of Donson?
Thank you. That's the next animation here. So the very top hit in the AlphaFold screen of Donson was Donson itself. The confidence metrics are off the charts, and we actually used mass photometry, as have a few other labs, to show that Donson actually does homodimerize. And it, of course, makes perfect sense, right? Because what Donson acts on is an MCM 2 through 7 double hexamer, right? So there are 2 copies of MCM that both need to be converted into CMG helicases. And so, although we didn't provide any evidence beyond showing that Donson can dimerize, we proposed that this preloading complex that we talk about, that consists of Donson GINs, top BP1, and pol epsilon, is actually a dimer, right? And that a dimer of preloading complexes docks or is tethered to the MCM double hexamer via those BRC treslin interactions that we just talked about, and that then this dimeric complex docks on. But there's a very interesting sort of twist here, no pun intended, and that is that if you try to dock a dimeric Donson onto the MCM double hexamer, it doesn't work. Okay, you can engage one MCM3, but the other MCM3 is in the wrong place. Okay, that's shown here, right? So this one interacts nicely, but then this loop is in the wrong place. And in order for it to bind, you would have to take the two MCMs apart, and one would have to rotate clockwise relative to the other, and that is actually precisely what the Diffley lab has shown happens during CMG assembly during yeast replication. And then a structure from the Costa lab of a frog CMG Donson complex actually confirms and very significantly extends that concept of separation and rotation plus some other structural changes.
So I also have two questions.
Yes.
First, can your database can be connected, for example, UniProt database, such as AlphaFold? And the second one is...
Sorry, I didn't actually understand the question. Can you say it again?
Okay, so the question is if your database of your data, the proteome or something like
predictomes,
that,
yes.
can be connected with UniProt database. For example, I open the protein and I can see the interacting proteins there.
Right, yeah. We've not done that yet. I think that will come. I don't know if it will be us who does that. I mean, if I should be so fortunate as to find someone who will give me $80 million to fold all the proteins, then I will certainly try to connect them to UniProt. But I think it's more likely that DeepMind or Google will be the ones to do this kind of thing. And then, no doubt, there will be all kinds of ways of accessing and triaging that data.
Okay, thank you. And the second question is, do you think that we can predict, for example, free proteins and find some other complexes that is forming triplexes or something like that?
Yes, so absolutely. We've done that. So a great example is another protein that we work on. It's called TRAPE. It functions at the it's an ether ubiquitin ligase. It associates with the replosome, and we used the same in silico screening approach to try to find partners. When we folded a monomeric TRAPE against all the replosome proteins, we found one interesting partner, plus TRAPE itself, which suggested that it homodimerizes, and there's evidence for that. So then we repeated the screen and used a dimer of TRAPE to screen against all of the other proteins, and then we got one additional very revealing hit. So that approach, and that protein was predicted to bind to the interface between the two TRAPE monomers. So absolutely, I think that's going to be a very important approach. Of course, doing that comprehensively, quickly brings you into sort of a combinatorial nightmare. So I think if you do all three ways, it's, I think, several billion. If you do all four ways, it becomes trillions. So I think for the foreseeable future, that will be a limited thing where you do it for a good reason. Only do it if you have a good reason.
Very nice. I'm just wondering, if you look at the structure predicted from the multimer prediction and compare them with the structure which are predicted from simple AlphaFold, do you think that these multimer prediction structures are more precise or more correct? Because you have actually complex, the whole complex, so which might maybe reflect more precise.
Sorry, you mean AlphaFold simple versus AlphaFold multimer, you mean?
Yeah. If you see the differences, in the prediction for a single protein in those two kind of softwares, and whether, if there is a difference, whether maybe the one which is in multimer is more precise.
No, I mean the only differences that we see are instances where the interaction of one protein with another leads to a change in the first protein. So the classic examples of that would be if you have an intrinsically disordered region, that has no discernible structure on its own, you fold it with an interacting protein, and now a little bit in the IDR of the first protein becomes an alpha helix. So we see that occasionally. But that's, I think, a function of one protein organizing another, and then not a difference in the structure prediction algorithms.
Yeah. So complexes with low Spock scoring, those which are experimentally determined, do they include PTMs?
Well, yeah, so maybe I overpromised in my talk. We don't understand yet what causes a low Spock score. So we literally, we have to go through and probably manually interrogate all the examples where there's a known complex that is scoring poorly, and perhaps even go into all the features and see which features were penalized. I mean, there's one, I think, sort of simple scenario, and that is, if you have two proteins that are part of a multi-protein complex, and their interaction is very, very limited, but they're scaffolded by other proteins, right? But by the criterion that we use, they're in a complex, right? Those are cases, I think, where AlphaFold just doesn't have, the interface isn't extensive enough for AlphaFold to have enough information to make the prediction. And so we're going to interrogate, one by one, all of the cases where things failed to see what fraction are accounted for by that simple idea.
Thank you for an excellent talk. I was wondering regarding your in silico screen, is there an intrinsic bias towards small proteins or small protein complexes? Let's say if you already have a monomer, the first protein that is 3,000 amino acids, will it be less likely that you pick interactions with such a protein than if it's 300 amino acid long protein?
Absolutely. So we've not done anything systematic in that space, but anecdotally, we see lots of examples where, as you say, you fold a huge protein with another protein, you don't pick up expected partners, and then what we routinely do is we segment the big protein and fold it. If we know what the partner is and where it binds, we'll just take that segment, and then often we will now observe the interaction. But if we don't know, we might actually tile the large protein and look at all of the segments. But I should point out that others, like Katja Luck's laboratory, has done this on a much more systematic scale. She had a nice paper a couple of months ago, and she showed that if you segment and use, I think, 100 to 200 amino acid segments is the optimum, then your sensitivity goes up, meaning you pick up more of the interactions, but your specificity goes down. So you start to see more junk, I guess. And I think one of the things that we would like to do is to see whether Spock can kind of clean up, those results and get us the best of both possible worlds in a segmentation scenario.
Right, but in the predictome database, this was not corrected, or no chopping was done there. It was just simply...
No. And in fact, you will see some conspicuous gaps in the data involving, two huge proteins that, when put together, simply exceed the amino acid limit of the GPU. For us, that's about 3,600 amino acids, or 4,000 technically. Yeah.
Thank you very much.
Thank you for a great talk. I think we'll really enjoyed it. I have just a question. I'm curious if you experienced any negativity bias when you trained your model because of most of the interactions. There was basically no interactions, and
So sorry, did you say negativity bias?
Yes, negativity bias for training the model.
Okay, and so this is where you expose my, fairly rudimentary understanding of computational biology. I was hoping actually to pipe my student Ernst into this presentation so that he could answer questions like that, but I'm going to have to. You can send me an email, and I'm happy to, have Ernst answer the question. But I don't think I have a good one for you. Yeah, sorry.
Okay, thank you very much for the discussion, and please join me again thanking Johannes for a fantastic lecture.